welcome back to the Catch Welfare platform, and I'm joined by my co-host, Michelle Boonstra. Michelle, how are you?
Yeah, very good.
So, Michelle, tell us what's been going on with the Catch Welfare platform since the last time we've been on air.
Well, mainly it's been holidays going on, but what's going on at the moment is that a lot of outreach is happening. So Catch Welfare platform is hosting a session next week at the ICES annual science conference. If I remember it correctly, it's session M. So Mike Breen and Hans van der Vis will be there, and a lot of our members will be there too, I guess. So stay tuned for that. I think it's available online. And in that same week, I will be hosting a session on fish welfare in China on the Animal Welfare Conference in Chongqing. Very interesting, I hope. And yeah, so that's mainly what's going on in this period with Catch Welfare platform. Our new team member, Lu Chen, is visiting Seafood Expo Asia today. So yeah. I hope she's having a good time there and meeting a lot of new members and existing members.
Now, today on our podcast, we will be speaking about catch welfare of pelagic fish. Particularly, we'll be talking about mackerel and herring. Could you tell us who's joining us for this podcast?
Yeah, so it's a busy podcast today. We got Lina de Nijs from the PFA. And we got Claus Sparrevohn from the Danish Pelagic Association. They're both core members of our expert group Pelagic. And they're joined by Mike Breen, of course, our own Catch Welfare team member. That has been doing a lot of research on pelagic fisheries and welfare.
So without further ado, we'll get to the podcast, and we hope you all enjoy listening to it.
Yeah. Lina, will you start introducing yourself and what you are doing working on welfare?
Yes, of course. My name is Lina and I'm working as a Fisheries Researcher for the Pelagic Freezer Trawler Association in the Netherlands. And we've been doing welfare research for a couple of years now, where I'll go out to sea and perform research on board the vessels to assess the welfare of the species coming in.
All right. And the PFA is an organization that represents various pelagic fisheries companies, right?
Yes, that's true. We represent the 3 largest Dutch fleet owners.
Yeah. All right. And Claus?
Yeah. Hi, everybody. My name is Claus Sparrevohn, and also a biologist like Nina. I'm a kind of a colleague to her, although I represent the Danish part of the pelagic fishing industry. And I've been working for them for the last 10 years, and one of our main focus subjects has been fish welfare for the last 2 years, I would guess. And it's we can see that it's a subject and topic that's picking up in Denmark quite rapidly at the moment.
Mm-hmm. Okay, and Mike, for the people that don't know you yet, just to be sure. Just to be sure.
There's plenty of them. Thanks, Michelle. I'm Mike Breen. I'm a member of the Catch Welfare platform, and I also work for the Institute of Marine Research in Bergen in Norway, which is a government science institute. So, and my interests are generally in terms of, it started with the behaviour and then evolved into the welfare of animals as they're caught in different types of fishing gears. But I have done some work on in both pelagic trawl and particularly in purseine with pelagic species. So that's hopefully what I'll be chatting about today.
Yes. All right. Lina, can you tell us a little bit more about the recent project that you've been doing together with Wageningen Marine Research? What it is about? What are you doing exactly, and what answers are you trying to answer with that study?
Yes, of course. So there's a lot still unknown about how, in what condition a fish come on board, and how they experience it in if you look at fish welfare. So there's a great knowledge gap there, and that's one of the reasons we're very motivated to look further into it. We started with just checking the condition of the fish when they came on board, but every time we go out, there's we have some additions. So we want to know during the whole capture and processing process how the fish are doing and how the conditions are doing, changing around the fish. So in the pelagic fisheries, we catch the fish in the net, and then the fish aggregates in the cod end. And we wanted to know what happens in the cod end concerning oxygen saturation and temperature changes. So last trip, I just came back 2 weeks ago. We looked into that specifically. So we had 2 sensors in the cod end to analyze the oxygen saturation there and how that changes over time during hauling. Then when the fish come on board, we have assessed the condition of the fish using the RAMP score method, reflex action mortality predictor score. Then the fish enter the fish tank. Of course, it's a tank with refrigerated seawater, so water from around 1 degree Celsius. What happens when the fish enter? So there we put in a sensor as well to measure oxygen saturation and temperature changes in the fish tank. And then later on, last research, we also looked at the pH measurements from fish and whether that differed between fish which died in net or fish which died on board. So that was our last trip where we've been working on, but quite some research and analysis are still ongoing, of course.
Yeah, right. Okay, so interesting. So, and I assume you try to relate those conditions like oxygen level and pH to the state of the fish. Are you just looking at the mortality, or are you also looking at the vitality? That's the RAMP score.
Yeah, we use the RAMP score, and for example, we try to relate that. Last year, we did a research where we had a sensor in the tank, and we used the vitality measurements on deck. And we saw that if the fish entered the tank, and there was quite a large proportion of fish entering the tank alive, we really saw a steep decline in oxygen saturation and a high rise in temperature. Then they started cooling, and it changed over time, but there was a really clear graph. While when we assessed that there was only a small proportion of fish entering the tank alive, so much more dead, we did not see that clear pattern. So that's how we try to relate it all together.
Yeah. All right. Claus, you are doing similar or not similar research. I'm not certain, but I think definitely when you guys are fishing with different methods, and you're looking at the different species as well. Is it not?
I actually would say that it's more complementary to what Lina has been doing. And of course, there are some differences in our fisheries, which is mainly related to what happens after the fish has entered the boat. Lina's vessels are what is called a freezer trawler or factory vessel, where there is a factory where the fish are being, after they're being caught, then they're being sorted in the factory and then frozen and sold frozen to different parts of the world. Whereas our fisheries are going directly into an RSW tanks. It's a tank boat, also called, where they are stored for delivery at the factories. So it is similar, but there's also small differences in the sort of volume and so on, and trawl lengths. But what we have done differently, I guess, is that we have been focusing. I should say that we have just finished our first part of our project here, and we are in between applying for another project. In the first part of the project, we wanted actually to go all the way and look for methods to have a humane slaughter on board the vessel. So when we did design the project, we had the idea that all the fish, or the main part of the fish, main bulk of the fish, would enter the vessel alive. Specifically in the herring fishery here, where we have conducted our experiments. But it turned out differently. It turned out that the vast majority of the fish on the 2 trips we examined, and 5 hauls, I should mention, there is large variety between species, but also within species. But for these 5 hauls, all the fish were dead when they entered the vessel. So we didn't really ever get a chance to sort of investigate how you could have a humane slaughter on board.
No.
Instead, we had a much more, and you could also say this ramp analyze didn't make much sense, because if the fish, if it's 100% of them, then you can try to get them to move as much as you like for a whole day, but it doesn't really give you much information on what's going on with them. So instead, we had to look much more into the hormone and lactate concentration, the more sort of the blood content of the fish, and that was in order to see what kind of stress level that has been experienced.
Yeah. So there's different ways to measure things. So just for people to sum up, because I get a lot of questions about this, you can measure lactate. You can look at the vitality, so behavior. You can look at mortality rates, and of course you relate that to environmental indicators like oxygen and pH and other things. So Claus, you've been looking at various things. What are the results? Do you have any results already?
We do have some results ready, and some of them has actually also been published. I think to sum it up easily is to say that we did see very high concentration of stress-related hormone, and lactate was mainly cortisol and lactate that increased quite a lot, and that did indicate that there has been a period of stress. We also found that the majority of the fish has lost all their scales. I would say all, I'm thinking 95% or more of the herring had lost their scale, which was maybe also a little bit surprising. But actually, when we compared to the control group we had in the study, which was rod and reel caught herring, we actually found that just catching them with a rod and handling them, putting them into a bucket, resulted in a loss of scales summing up to 67%. So maybe it's not that surprising that it's 100% in a commercial trawl where it's going back through the water. So they're very sensitive to this.
Yeah, and how? Because I can imagine when you are in contact with gear and you're with thousands of other herring in a net, I can imagine you lose your scale. But without that, just by rod and line, that does surprise me actually. Does anybody know how that happens? Do they just throw them off?
No, there's I think it's rod and line and handling. So it's also when you take the fish and get them off the hook. It's probably there the most
at that moment
scale loss takes place.
Yeah, herring have what are referred to as deciduous scales. So they're very large scales relative to their body size, and they're rather loosely held in sort of the skin of a fish. There's multiple layers. So the outer layer is a membrane, and then you have the scales, and then you have a dermis layer. In which there are pockets which the scales grow out of, very much like our fingernails grow out of sort of pockets at the end of our fingers. And with herring, the membrane is very thin. Membrane is normally very thin in most fish anyway. But the scales are loosely held, and it's been hypothesized that actually the easy shedding of these scales, which are very reflective, could be a defense measure, particularly in the schooling fish. So it's almost like chaff being spread out of an aeroplane or a ship, sort of an anti-missile defense. So you send out these scales which go fluttering down through the water, which visually distract any predator. it's a hypothesis. But particularly with herring, so they very easily lose their scales, very easily. I mean, you just touch the skin of a herring, and your hand will be covered in scales.
That's a very interesting theory. It's a little bit like an octopus does, right? Confusing.
We think. Yeah. So it's an interesting defense mechanism. But so from that point of view, we need to be careful about how we interpret just scale loss on its own.
Yeah.
So the scale loss in itself might not actually be that traumatic or stressful for the fish, in that if it's part of a defense mechanism anyway. No. What will be damaging and what will compromise the physiology of the fish is if the lower levels of the layers underneath the scales start to become heavily damaged. So that's the dermis and the sort of the connective tissues underneath the dermis that link it to the muscle. If that starts to become damaged, then the fish will have problems. It's then exposed to pathogens, and it will be exposed to tissue loss from the underlying muscle. So that could be a real problem for the fish. So it's what's really interesting is, yes, these fish have got a lot of scale loss. What will be interesting next to look at is, okay, how deep does that skin damage go in terms of that, in terms of those multiple layers within the skin of the fish. So really interesting. I mean, I congratulate you on the paper which came out recently, Klaus. It was
Thank you.
Very interesting to read. I've enjoyed seeing this project develop. It's nice to see this work getting published now. So congratulations and keep up the great work. And Lina, when are you publishing?
Could I just, before we go on, I have a question for you, Mike. I seem to remember that Maria Tenning did some experiments with removing some scales from herring. I don't know if you were on that work.
I wasn't directly involved, but my colleague Maya Tenningen, she did that as part of her PhD. Yes.
But is it correct that she didn't see any effect on the mortality when she removed up to 25% of the?
Yeah, there was no significant effect. And again, they didn't really go into detail about how deep they were going in. They were essentially trying to just remove scales. So yeah, it was. So we don't really know how deep the penetration was in terms of the damage. So again, it's difficult to interpret. But yeah, with 25% scale loss, if I remember correctly, I'd need to go and double check. But there was no significant effect on mortality.
Yeah, that's the same I recall.
Yeah. How quickly were you catching the fish before the blood sample was taken?
With the run with the inline, that was very quickly. That was within a minute, I would guess.
Oh, you've got a very good chance of getting a nice baseline.
Yeah. One thing we hadn't touched upon is also that we did find, and that also brings us a little bit back to the scale discussion. We did actually find that a lot of the fish caught on the commercial vessel, on the trawler, showed sign of hemolysis, which basically mean that the red blood cell had starting to dissolve. And we do sort of wonder if that's something that it probably happens before death, or no, I'm not sure whether we actually were able to conclude when exactly that happened. But it's something that we would like to look more into and to see if that's, for example, related to the scale loss, which we originally thought that it might be, that there might be some osmolysis, mortality problems, or whatever it was. But it's interesting, and it has some aspects related to quality of the fillet as well. So it is also from a commercial point of view worth looking into why this happened and when it happens in the fishing process.
Yeah, talking about quality, is that something you're looking at specifically, Lina or Klaus?
Well, of course, it's an important factor for commercial fisheries because the fish is for commercial consumption, human consumption. So the quality is very important. But there's not much knowledge yet on how quality and welfare for the fish relate. So that's one of the reasons why we're looking into it with our research.
So you look at that
with the pH measurements.
Yeah, so you're looking at that specifically. Mike, you've been looking at the relationship between quality and welfare, I think also for pelagic species, right?
Yeah, particularly for mackerel. So this was mackerel caught in purse seines. So we did some controlled experiments, simulation experiments. So one of the main stresses in the purse seine capture process is crowding. A purse seine is a very large net, and it surrounds the school. And then over a period of about 3/4 of an hour, that net is gradually brought in to crowd the school next to the boat. Where in our fishery, in the Norwegian fishery, we use pumps then to pump the fish aboard. Now that crowding process is stressful, very similar to the crowding process which Lina and Claus were talking about in terms of the cod end. So the fish are crowded together. There's not much water to biomass, so there's potential for reduction in oxygen levels, which Lina was looking at, and we're going to revisit that later on. So you've got potential for hypoxia, asphyxiation, but just being crowded is very stressful for the fish. So what we did is we did some simulation experiments basically in aquaculture cages with schools of mackerel, and then we crowded those fish for different lengths of time, and then we took those fish under controlled conditions, and they were then processed, so filleted, and those fillets were monitored instantaneously for quality and chemistry after a few days, and then after a year in deep freeze, and over a series of experiments. So this was sort of multiple projects, but with all the data combined, what we saw is that very stressed fish certainly had an impact on immediate quality, and we could see an increase in sort of softening of the fillets, an increase in a change in colour in the fillets. So there was more red, which relates to sort of increased blood content in the fillets. We saw a greater probability of gaping, which is when the fillet begins to separate between the muscle blocks, which is a standard indicator of reduced quality. But then looking at the chemistry, particularly after 12 months storage. Now, got to be absolutely correct here. The storage has the major effect on the quality of the fillet and the chemistry in the sort of the meat chemistry. By far the most important factor is storage. But what we could also see after 12 months, we could pick out whether the fish were from the control group, a low stress group, or a high stress group. We could still see that signal. So the stress was basically accelerating the decomposition in the freezer, essentially, which is really interesting, because that then leads to questions about storage and shelf life.
Yeah,
which is why. Yeah, it's why the fish buyers are now becoming interested. So the likes of Marks and Spencers and Tesco's are now becoming interested in the importance of pre-mortem stress, so the catch welfare, and the link with shelf life. Because this all ties up with sustainability. This is not just sustainability is often thought about how heavily you harvest the stock. If you've got bycatch, any animals you're releasing, do they have the opportunities to survive? That's sort of the traditional look at sustainability. But also, if through poor welfare we're reducing shelf life, then that means we're potentially increasing wastage, which means we've got to catch more fish to replace the fish that's been wasted. So this all loops back to sustainability. Yep. So it's the same discussion at both ends of the supply chain.
Yeah. Yeah. So that's important for people to realize. It's not just about the welfare of fish and the ethical aspect, but it's much more. So it's a very strong indication if you can see the difference after 12 months in the freeze.
Yeah, there was a clear interaction. There was a clear interaction in the data, and it wasn't just, we weren't just looking at one parameter. We were looking at, I can't remember, I'd have to go back and look at the papers. I think we're looking at 15 different parameters in the mussel chemistry. And of those 15, I think we saw significant interactions related to pre-mortem stress in at least 5, I think maybe 7 of those parameters. So it wasn't just a one-off hit. It was we're seeing a consistent story.
Wow. All right, so yeah, quality and sustainability could be reasons to work on welfare. Lina, what was for you the reason or for PFA to start working on welfare?
I think that it was before my time, before I worked at the Pelagic Freezer Trawler Association. It was when I was still a student. And then the PFA got a questions, Wageningen Marine Research, like, can you help us with this kind of research? I think it was because there's increasing societal attention for this aspect, because we're all thinking about the chickens and the cows and the big stables, but you see increasing attention like, oh, but the fish on my plate, what happened there? So that was one of the reasons, but also there's like a big knowledge gap, and there's been talk about stunning fish and humane slaughter on board. But before you can implement something, you first need to assess what the conditions are. And we didn't know that. So that was one of the reasons, like the big knowledge gap. First, there needs to be a scientific basis. You need to know what's happening before you can look into possible solutions or improvements. So that's why we're working on it quite actively at the moment.
Yeah, clear. Yeah, no, that's definitely true. You first need to know what we're talking about.
Yes.
Yeah. And Klaus, how was that for you? What were the main reasons to start working on this topic?
The main reason was that we were approached by a consultant working for Animal Protection Denmark, who had recently been hired to look into fish welfare.
Mhm.
And he did what I think was a clever idea from his side. Was he looked into how many fish are actually involved in, for example, aquaculture in Denmark, how many fish are involved in the demersal fish, how many fish are in the aquarium back at people's home, and then he looked at how many fish are being caught in the small pelagic fishery and industrial fishery. And I can assure you, I don't think that the numbers in the pelagic fishery is, of course, much higher.
Yeah.
And then he said to himself, why don't we look into that to see if we don't even probably need to make a big difference, but if we can even do a small difference, we can do a small difference for a lot of fish instead of focusing on the conditions of a few fish.
Yeah.
And then he approached us, and we have a strong tradition from being involved in all kinds of scientific projects, also projects that we sort of can see might be sensitive to our businesses, but nevertheless we engaged with him.
Yeah. So if people are talking about numbers, then they soon end up at your doorstep.
Yes.
Yeah.
That's for sure.
And what were the reactions of your members when you came up with this idea?
I think I can say that all my members think it is, to be saying directly, find it a bit ridiculous to look into this, to be honest. I think that they, in their mind, that if you talk about animal welfare, there's enough problems in, for example, cultured cows and pigs and stuff like that. But we also tell them that it's not their own personal opinion that's really driving their businesses. It's of course the end consumer, and it's the market. And if the market wants them to look into it, if the retailers wants them to improve on these issues, then they will do it. They are professional people, and they're doing it fully, and are engaging in collecting data and discussions. But at a personal level, I also must say that I think a lot of them do think it is a little bit strange to look into.
Yeah, that's what you hear more often. But yeah, in the end of the day, fisherman is not catching fish to consume it all by themselves. You catch it to sell it to somebody else, and then what they expect and think is important matters as well, of course. That's how the market works.
That's exactly why we started our work, looking at trying to link pre-capture stresses with quality, so that we could actually have an honest conversation with the fishermen.
Yeah.
And it was actually speaking to some skippers who I noticed were taking particular care over their purse seine catches, trying not to overcrowd them, particularly with mackerel. And when I spoke to them, they said, "Well, we don't want them to die in the net. We want them to die in the RSW tank. We don't want them to die in the net. If they boil, which is when they start swimming crazily to try and escape." Yeah. And so the water just literally boils.
Yeah.
All you can see is mackerel, and that doesn't necessarily have to happen. It can happen with some skippers all the time, because that's how they fish. But the better skippers try to crowd it to a point where they're not boiling the catch, and then start pumping, and then all of those mackerel, and the objective is to get those fish alive into the tank.
Yeah.
And you ask the question, well, why are you trying to do that? And they said to me, the quality's better.
Yeah.
And
So they have that knowledge already.
Yeah,
Actually.
those skippers tend to get a better price.
Yeah.
Now,
so they try to prevent that point where the mackerel start to panic.
Yeah.
Yeah.
So those skippers who tend to get the better prices are already conscious of welfare practices. They would never refer to it as welfare. It's just how they're handling their catch. But you can have a grown-up conversation with them and say, they're skeptical about the degree we're looking at. So for example, exactly like Lina and Claus wanted to do, but Lina did when she was looking at the conditions inside the tanks. We did the same with the purse seine catches, and we showed that with mackerel they do tend to die quickly. We see a drop in oxygen levels in the tank, but it's not very rapid, and we're seeing a loss of vitality within about 30 minutes. And that's more to do with the fact that mackerel muscle, swimming muscle, really doesn't function below 5 degrees. So they can't swim, so they tend to sink to the bottom of the tank. And then when we do an oxygen profile, we can see that the oxygen at the bottom of the tank is very low indeed. So they are dying from asphyxiation in the tank, and that's something we need to look at into more detail. That's going to be stressful for the animal. Is there a way of improving that so we can maybe improve the quality even further, improve the shelf life further? And that's an unknown quantity at the moment. That's something we'd like to look at. With herring, what was interesting, and this relates exactly to what you saw, Lena. We never got the control over some catches were mostly dead, some catches mostly alive. With the herring, all the catches being pumped in were mostly alive from a purse seine. And that's pretty typical. But what we did see, we tried to keep catches separate in different tanks. So where we had a large catch, that would mostly fill a tank, or fill it to the optimum level that they would have for storage. And what we saw in those tanks is we saw a very rapid drop off in oxygen, and we saw a corresponding drop in vitality. So how alive the fish is, sort of different behaviors switching off. And we saw that occurred at about, well, once the levels got below about 2 milligrams per liter of oxygen, which equates to about, at those temperatures, I think equates to about 20%, 15-20% saturation. Once it started dropping below there, then we saw a rapid drop off in vitality. Now, what was really interesting, so these tanks are held at a temperature, it's below freezing, but because it's salt water, it doesn't freeze. So initially, when the first fish going into the tank are going into water, which is at about -1 degrees. That temperature goes up as the warmer fish are coming in. And it'll go up to 2, 3, maybe even 4 degrees, depending on how much catch is going in. But it's typically below 5 degrees. Now, where we had small catches, we saw a much lower drop-off rate in oxygen. And those fish could survive in those very, very cold temperatures for 6-8 hours before we started seeing a loss in vitality. And again, the loss in vitality was correlated with the oxygen level in the tank. So the herring are also dying of asphyxiation, but because, and what we do know of herring, in particularly sort of in northern Norway, they are adapted to cold weather conditions, cold water conditions. So they overwinter in fjords, and they form fairly thick layers in fjords, and they can survive at temperatures 2-3 degrees quite comfortably. So they're already adapted to cold water temperatures, so they can swim around the tanks no problem. So for them, it's the hypoxia, which was appears to be the main killing factor.
So can you conclude from that smaller catches increase the welfare and gives higher vitality, or is that not? Is that
well,
too
it would
conclude?
It would during crowding conditions, but no, that's not. We were deliberately keeping catches separate, right? That's not necessarily typical.
No.
So a fisherman would tend to try and fill his tanks up. Now, if there was a few days between catches, he or she wouldn't put a catch, a fresh catch in a tank 3-4 days old. That would be unusual. They would try and keep catches together. They might do. It depends on the fishery at the time. But if a tank was only partially filled, the chances are the herring would live for several hours. Whereas the tank was full, and they moved on to another tank, then those fish would tend to die quickly. So it's more to do with how full the tank is as opposed to the size of the catch. So, but if you only had a small catch and all your tanks were empty, then yes, your fish are going to live longer. Now, what impact that's having on other stress parameters, we don't know, because we weren't looking in detail at the physiology at the time. We did look at lactate. Lactate did increase over time, but we'd like to look at it in more detail and relate it to more detailed physiological parameters and more detailed meat quality and meat chemistry parameters as well. And that's, again, we need more funding and the usual story.
More research is needed.
Yeah, sorry, it's
as usual. But it comes down to you're trying to reduce the stress or mortality in the process. But in the end of the day, then they are more alive in the RSW tank, but they die sooner or later. Is it worth to improve the welfare during the capture if that means they will die then in the RSW tanks? Or is that only worth doing that if you apply a stunning method? Because we've been having that discussion before, and it is something that.
But if we think of the whole capture process, right? Mm-hmm. There are very few capture methods which are benign, which are non-stressful to the capture.
Yeah.
Right. So, and a fundamental principle of good welfare practices is if you're going to stress an animal, you try and keep that stress level as low as possible, and you try and keep the exposure to the stressor as short as possible.
Yeah.
Okay. Now, at the moment we don't really have a great deal of control over the level of the stressor. So what we'd be looking at is trying to reduce the exposure times.
Yeah.
Now, so in principle, if the fish are dying quicker in the tank, the degree of the stressor may be higher, but the exposure is shorter. And what we need to look at is really what impact is that having on the what impact is that having on the blood chemistry and the muscle chemistry, and so we can have a better understanding of how that's going to affect sustainability. Okay, yeah. All the animals in the catch you're keeping are going to die, so we're only interested in mortality in animals we want to release. All the animals are going to die quickly in the catch, or are going to die in the catch, and so in principle, the sooner they die, the better, because you minimize an exposure.
Yeah.
And then ideally, then you can begin to control. You can begin to control the magnitude of that stressor, and so get the stress time down to a minimum period, and try and reduce the actual stressors, and then you're in an optimal situation. But we're really just learning about these processes now. Yeah. We don't know what different stressors are having on the animals. We don't, and one of the easiest things to measure is the exposure time to the different stresses.
Yeah,
and that's some of the things that Claus has been focusing on, Lina's been focusing on, and my colleagues, and we've been focusing on, and trying to understand that better.
Yeah, so there's different ways of improving welfare.
That's one of the reasons why we are a member of the Catch Welfare platform, because we are assessing the situation and several parts of the catching process, but there is a need for discussions in international fora like the Catch Welfare platform. What is considered to be a welfare benefit for the fish? Because those are fish for human consumption. They're gonna die anyway.
Yeah.
So there needs to be, we need to find out actually what would be the best way to see what's happening, but what's considered to be a welfare improvement.
Yeah,
that's still in discussion, I think.
Yeah.
But what we saw is that it's very fishery dependent. The survival of the catch and the mean vitality score is really influenced by, for example, trawling time. Species is very important, but also the size of the catches. For us in pelagic trawling, you see a really clear statement that the smaller catches come on board much more lively than the bigger catches. So
yes, maybe that's even more important than whole duration, or is it not? Can you not compare them like that?
I do think it is, and whole duration is sometimes also a bit difficult, because maybe you're trawling for 4 hours, but the catch only comes in the last half an hour.
Yeah, absolutely.
So that can be also quite a difference. But I saw last trip when I was on board, I saw catches from 5 or 10 tons. And I saw a catch which was a few times bigger than that, over 100 or close to 200 tons. And we really saw a great difference in vitality.
Yeah. So that's a difference with, for example, demersal fisheries, where fish come into the net more, spread out during the whole. Of course, you're targeting schooling fish, so it's one moment they'll enter the net. Yeah.
Yeah, but if you see a school on the echo center, it's like, oh, I'm gonna put my net out. If you miss it, then you have to turn around, and it will take some time. And you're trying again, and maybe you catch it, and maybe you don't, and then you try another school again. So it can be, the trawling time can be quite high, but that does not mean that the fish spend that much time in the net.
Yeah, that's definitely true. Yeah, that's why it's everything is so gear specific.
It is.
Can I ask a follow-up question? So when Lina was introducing herself and introducing her project, she mentioned measuring oxygen in the cod end. Now that's particularly exciting, because it's something I've been trying to do for years. So firstly, congratulations. I think you're the first. So you should be getting that published. Okay.
We're really trying our best.
Good. And we were chatting offline, so I know Lina's got some results, just some preliminary results. Could you tell us a little bit about sort of what oxygen levels you were seeing inside the cod end, and that really does follow up from what you were saying in terms of it's when the catch enters the cod end, So I'm looking at analysis of exposure to low oxygen levels. So anyway, you tell a bit more about the oxygen results.
Yes, I will. So we put in the two sensors, one in the middle of the cod end, kind of like a spider, and one on the side of the cod end. We only did this 3 weeks ago, so it's really still preliminary, but we made some first graphs, and what we saw was when we're going into the water and we reached our fishing depth, the oxygen saturation was already at 80% instead of 100.
Mm-hmm.
So we also need to check our sensors whether they're like correcting for the temperature change, which was there. But what we clearly saw was we noted down when the different catch sensors came in. Those are sensors which are being placed on the net, and they are kind of signaling back to the boat, like now you have so many catch in your net, now you have so many catch, and we knew where our sensors were being attached. So and then we tried to relate it to the time to see if you can actually see how many fish is there in the graph of the oxygen saturation. And it was quite clear there. So what we saw was that the oxygen saturation was going quite low, and you really saw different steps on the way where there was fish coming in. And during like the last part of the trawling time, we saw oxygen saturation levels of 5 to 10% only from the sensor, which was in the middle of the fish. So it was just covered all around with fish, and there it was quite low oxygen saturation. We had a camera placed as well there, and we saw that the water flow in the cod end wasn't very strong either. And you really saw a difference when we were coming up back to the surface. You have some things during hauling, like picking over from the fishing boards. You pick them back to the boat and stuff. Those are moments that the ship is going really slow. You really saw the fish swimming up back to the front, where the sensor actually got some fresh water again. So you really see the peaks while moving with the vessel and different stages during hauling. So we're really curious how the other graphs will look, but it was very interesting to see that it really goes down to very low levels already during the fisheries.
Yeah, that's makes a lot of sense, because there really is very little. Once you get a very crowded biomass, there's very little water in between those fish.
Yeah.
So they quickly strip the oxygen out of the water. We see the same in the purse seine and in our crowding experiments. We don't see levels as low as that, though.
We're seeing different vitality levels or stress responses from different gears. How can that be interpreted?
Right, so you're referring to sort of the potentially different stress levels and vitality levels in the catches we're seeing between the different fisheries. I think one thing we have to be careful about is sort of over-interpreting or trying to identify an ideal capture method. So different capture methods work in different ways using different behavioral responses, and yes, they're likely to produce different stress responses, but these different fishing methods have evolved in fisheries for very practical reasons, and so they've evolved in some cases over hundreds of years. And the capture method it's a complex interaction between the technology, the behavior of the animal you're trying to catch, and the environment in which it's being caught in. And so, for example, it might be easy to say, well, all the catches coming in alive from purse seine. Why don't you just use purse seine to catch all the pelagic fish? Well, purse seine only works in specific circumstances. You've got to have good schools. They've got to be of a particular density. Otherwise, it's just not worth using purse seine. And you've got to have weather conditions which allow that to be used. Whereas pelagic trawl is far more generally applicable and can be used to catch fish in much lower densities. And so we really need to be careful about how we overinterpret our results. So it's not like we're presenting an ideal fishing gear. What we're trying to do is understand the capture processes in each fishery, and then try and improve them individually. So we get we improve welfare, and we improve the quality of the fish that's being produced, and hopefully we increase the sustainability. So that's our strategy at the moment.
Yeah, I very much agree with what Mike just said. Yeah, that this diversity of the fishery is subject that's really difficult to sort of communicate. My experiments with the paper we just released was that we did a press release on it, and immediately after that, people were sort of concluding that based on our experiment, which was 2 trips and 5 hauls in the North Sea herring fishery, then the conclusion was that for all North Sea herring being caught in the entire fishery, all of them were dead. And I think this conclusion is really, really problematic to take. I think you have to look at from fishery to fishery, season to season. Small gear modifications might also mean quite a lot, and I think that is really difficult to communicate to journalists, for example, because they like to have a simple answer to a complex question.
Yeah, that's the case in with more subjects, that people always want the simple story,
Yeah.
but it's always more complex than people think.
Hans and I are involved in a project at the moment, an EU project, where we're looking at these animal-based measures, as sometimes they're referred to, these behavioural metrics, and sometimes physiological metrics as well, and trying to relate them to EEG, electroencephalogram readings, and ECG, electrocardiogram readings, and so from the EEG and ECG combination, you can determine whether an animal is actually dead or not.
Yeah,
that's the only guaranteed way of looking at it. And actually, even then, the only guaranteed way of knowing an animal is dead, and we've had long discussions over this, is when you see it start to decompose. Right? It really goes back down to that level, but that can take 24 hours, right? Which is we can't monitor each animal, and part of the point of this project is to come up with correlates of dead, of lack of EEG activity for a prolonged enough time to know the animal is brain dead. To relate it to some of these vitality indicators. I mean, some are pointless. Some we know. For example, just the the body flex that you'll use within your ramp score. So when Lenny gets a high score, when we use vitality, we get a low score, and that's something we need to resolve actually. But that's a different conversation. We need to standardize how we talk about vitality, because otherwise it's going to be confusing for viewers. But some metrics are clearly, so for example, body flex. So you lay the fish down and it starts flapping. Now the fact that it's not flapping around on your measuring board or whatever, really does not relate. It's not a high probability indicator that the animal is dead, because we know from the work I've been doing recently, we know that tends to disappear fairly early in the sort of downward slope of vitality, right? But some of the last things to disappear, like the sort of ventilation response, the head complex, so it moving, it trying to breathe, and I think the ultimate one, which is the eye roll, the vestibular ocular reflex, which is when you turn the fish side to side. It will try and, if it's alive and its brain is functioning, its brainstem is functioning, even if it's not conscious, it will try and maintain its eyes level with the horizon. So you see its eyes roll. Okay. Now, when we know that is that's mediated in the brainstem. So if
Yeah.
that's not working, either the nervous link to the eyes, the eye muscles is broken, or the brainstem is no longer functioning. Right. So the VOR, the eye roll is going to be a really good candidate, I think, for a correlate to brain death.
Yeah, and
But we
that
need
needs
to
to be validated by.
and that's exactly what Hans and I and others, including Albin,
Okay.
that's exactly what we're working on with a whole range of different species, and we're also going to be using physiology to try and confirm this as well. So there's multiple metrics we're going to be looking at over the next 3 years.
Okay, now we're going to do it on record. Try to
Try to make it concise. I know.
And because so far I think it's been pretty scientific. So try to avoid matrices and these kind of.
Yeah.
Well, yeah. Try to keep it as simple as possible. So what was the question again? Okay. Yeah.
How do we know the fish are dead?
Yeah. So during this discussion, we've been talking a lot about fish being dead or not. But when you're looking, when you're doing vitality tests and fish appear to be dead, the question is the fish really dead, and how do we know? Of course, the EEG method is known to be the very, very proof of a fish is dead or not. Mike, how is that in this type of studies, and what is needed?
What Lena, Klaus, and I have been looking at, with our colleagues, what we've been looking at with when we refer to vitality or ramp, what we're looking at is a collection of different behaviours or different reflexes that the animal have, and essentially what you do is you have a test to try and trigger a response from the animal, and it's a standardized test. There's a particular protocol for each test, but these are done very quickly. These can be done in like 30, 60 seconds, as in the whole vitality assessments, and you would typically have somewhere between 5 and 10 different metrics or different assessments within that overall assessment, and you're basically looking for a reflex or a behavioral response or not. That's fundamentally what it is. These methods are still being developed, so if I see a behavioral response, I'll score a 1. And then you've got a time frame in which you can score a full response, and it's typically around 3 to 5 seconds. Okay, and if that behavior doesn't come in that time, then it gets a 0, or you might get a weak response you're not sure about, and then it'll get a half, outscore half. Or if there's a delay, so if we're looking at another response, or we haven't started looking at another response, but we get a reaction after the 3 seconds, then it'll be noted as a weak response. Now, if you get even a weak response, then the animal's not dead in any one of those parameters. Now, the question is, if you get no response in all of those, is the animal dead? Well, we still don't know, because there's work ongoing at the moment as part of an EU project that Hans and I and others are involved in, where we're looking at these behavioral reflexes, and we're trying to relate them to both brain and heart activity, and to see using brain and heart activity as an indicator of when the animal is dead, or at least the brain and heart are not active for a prolonged time.
Yeah.
So if we can relate any one of those behaviors to them, then we've got a good correlate.
Yeah. So you validate those fatality with EEGs. But at this moment, when we talk about fish being dead during the study, it might well be that they are still conscious, but just immobilized and unable to respond.
Well, I can't comment on the consciousness in terms of sentience, because we don't. There's more and more evidence coming in to suggest that animals are responsive to different inputs, and we get a complex electrical brain activity, which could correlate to consciousness. We don't refer to it as consciousness. We refer to it as electrical brain activity, and we're looking for coordinated responses in that activity as well. And that's essentially our baseline. That's what we're looking for. These complex responses, we're not really referring to it as consciousness, but if there's no brain activity, there's no potential for consciousness if consciousness did exist in these particular species.
That's for sure. Yes.
Lina, in your research, how do you assess if a fish is dead or not?
So we're using the RAM fertility score to assess the fish when they come on board. And then you look at specific reflexes, but the fish need to perform them within a certain time period. Depending on whether they show the reflex, yes or no, they're scoring a point, and they get eventually, after a few, I think 9 reflexes, they get a final score. However, if the fish does perform the reflex, but it does it outside of the set time period, the fish is not dead. And then in that case, it can score a really low score, 0, because it doesn't perform all the reflexes. However, it may have reacted afterwards, after the set time period, and then it also gets a score like seems to be alive. However, if the fish does not do anything when looking at it and does not respond at all, then they get like a specific score for seems to be dead. But it always seems to be, because we do not check it with any brain activity or heart activity. But it's just a feeling in that sense, an observation.
And going forward with catch welfare and its research, how important will it be for all researchers and the different areas to come together and create a general score card so that everybody can reference back to it and understand what everyone's talking about?
That's very important, because between the Dutch research and the Norwegian research, we used the score exactly the other way around, and we had quite some funny moments where we misinterpreted the results from each other. So it's really important that there's just going to be one method where we score everything the same way, just to make everything clear.
Well, we'd like to thank you all for joining us today, and it was a very interesting chat, and we'd like to hear more about your research in the future, so we'd be happy to have you back here.
Thank you so much.
Yeah, thank you, Oliver. It's, as always, it's a pleasure chatting with you. So I've really enjoyed today. Thank you.
Yeah, see you all. Bye-bye.
Bye.
Bye.
Bye.